Hi, welcome to the SUMOylation lecture on your advanced techniques in molecular biochemistry. I am Dr. Germán Rosas-Acosta, and my research interest is on the regulation of the cellular simulation system, specifically how the cell is capable to modulate global simulation levels, as well as the interactions that are established between the simulation system and viral infections, specifically making a special emphasis on the Influenza A virus. For this first lecture of the simulation component, I will be dealing with a brief introduction to post-translational modifications, followed by an introduction to SUMO and the SUMOylation system, followed by the functions and relevance of SUMOylation, not only for proteins, but for the cell in general, and I will end with a brief discussion on some of the critical issues and questions that are related to the simulation system. So first, let's get started with post-translational modifications. And for this, it's important to keep in mind that the functions that are associated with different cells in different tissues are all directly determined by the proteins that are expressed in every different cell in your body. And when it comes down to those proteins, the functionality of each one of the proteins that is expressed in the cell is modulated or dictated by two different properties. The first one, the shape of the protein. The shape of proteins dictates very closely the kind of functions that can be associated to that particular protein. The second property that is key in dictating the functionality of different proteins within the cell is the location of those proteins in the cell. And both of them, the shape and the location of proteins within the cell, can be modulated. And that modulation is typically achieved by post-translational modifications. There are many different types of post-translational modifications, and many of them you have probably heard about them in other classes, such as in molecular cell biology. A post-translational modification can be thought of as any change in the chemical composition of a protein, where it is by deleting specific segments of the protein by cleavage or by attaching additional chemical groups or entire peptides to the protein. To be considered a post-translational modification, any chemical group that is being attached to the protein must be attached through an actual covalent bond. There are numerous types of post-translational modifications that have been characterized, and some of them are actually shown in this slide. And among those, you have acetylation, methylation, farnesylation, phosphorylation, palmitylation, glycosylation, cleavage, myristylation, ubiquitination, and the one that corresponds to this lecture, SUMOylation. And notice how each one of those post-translational modifications happens through very specific amino acid residues within the sequence of the protein. Perhaps the most complex type of post-translational modifications is the attachment of a member of the Ubiquitin-like family of proteins to a protein. The Ubiquitin-like proteins, or UBLs, share as the main characteristic a very similar three-dimensional shape, referred to as the beta-beta, alpha-beta-beta, alpha-beta ubiquitin fold. In this tertiary structure, the alpha indicates alpha helices and the beta indicates beta sheets. And that sequence of beta and alpha structures is the main characteristic of all members of the UBL family of proteins. One additional feature that is shared by all members of this family is the presence of a diglycine motif at the C-terminus of the mature protein, and that diglycine motif is only exposed upon the processing of the immature protein. Additionally, that diglycine motif is the region that allows the conjugation of the ubiquitin-like protein to the target protein. One final feature that is shared by all of these proteins is that conjugation always takes place through a lysine residue located within the target protein. The best known member of the UBL family of proteins is ubiquitin. And ubiquitin is most frequently conjugated to target proteins not as a single peptide of ubiquitin, but instead as a long chain of ubiquitins, something referred to as a polyubiquitin chain, in which one ubiquitin molecule is also ubiquitinated, and that ubiquitin is in turn also ubiquitinated with another ubiquitin, forming long polyubiquitin chains that then target the protein that has been polyubiquitinated to the proteasome, which is a multi-protein structure that degrades proteins within the cell. So polyubiquitination is typically thought of targeting proteins for proteasomal degradation. Importantly, the conjugation of ubiquitin to target proteins involves 3 different enzymatic stages: the so-called E1 enzyme, the E2 enzyme, and the E3 enzyme, also known as the ubiquitin ligase. This one is the one that confers specificity to the whole process of ubiquitination. SUMOylation involves the attachment of SUMO to a target protein, and similarly to ubiquitination, it also requires 3 different enzymatic steps: the E1 or activating enzyme, which is a heterodimer formed by the SAE1 and SAE2 proteins. The second enzymatic step is the conjugation step, which is mediated by UBC9, which is the only enzyme capable of conjugating SUMO to target proteins in the cell. And the last stage is the ligase, which for SUMOylation acts in combination with the E2 activity. So E3 enzymes or SUMO ligases can confer also specificity to the UBC9 conjugating enzyme, therefore facilitating the conjugation of SUMO to a very specific range of substrates. After SUMO is conjugated to a target protein, SUMO can be very quickly deconjugated, therefore leaving the target in the unstimulated form and producing free SUMO that can be used in another cycle of SUMO conjugation. The deconjugation step is mediated by SUMO proteases, which are also involved in the initial processing of the SUMO precursor protein, and that initial cleavage is absolutely required to expose the diglycine motif that is located near the C-terminus of SUMO. Without the processing, the precursor form of SUMO cannot be used for SUMO conjugation. So the initial cleavage of the SUMO precursor by the SUMO proteases is an absolute requirement to allow any new molecule of SUMO that has been synthesized in the cell to become active. Now, in sharp contrast with ubiquitination, which is typically associated with protein degradation by targeting proteins to the proteasome, SUMOylation affects protein function in many different ways. Among others, some of the effects that are produced by SUMOylation include changes in the stability, the solubility, the structure, and the localization of the target that is being SUMOylated. Furthermore, SUMOylation can also regulate the ability of the protein to be post-translationally modified by other types of post-translational modifications. One potential effect that SUMOylation has been postulated to exert that actually explains pretty much all of the other potential effects that had been described previously is the ability of SUMOylation to alter the profile of interactions that a given protein is able to establish within the cell. According to this model, once SUMO is attached to the protein, it typically triggers a slight change in the actual shape of the protein so that that particular protein that has been SUMOylated can now interact with other proteins that were not able to interact with the unmodified form of the protein. Once those interactions have been established, SUMO can be released, can be deconjugated from the protein, and still, even after SUMO is eliminated, the protein-protein interactions that happen in the presence of SUMO remain because the shapes that were adopted by the protein allow for those interactions to continue even in the absence of SUMO. And that is the reason why SUMO can have long-term effects, even though SUMOylation itself is a very transient type of post-translational modification. To understand the effects made by SUMOylation, we're going to now look at the specific effects made by SUMOylation on 4 different protein targets. RAN-GAP1, the RAN GTPase activating protein 1, was the very first protein known to be SUMOylated. And in fact, the discovery of SUMOylation as a post-translational modification was associated to the discovery that RAN-GAP1 was SUMOylated. For this target protein, RAN-GAP1, the initial studies looking at SUMOylation were able to determine that the residue in RAN-GAP1 that was targeted for SUMOylation was lysine 526, and mutating that specific lysine residue into an arginine residue completely abolished the SUMOylation of the protein in both an in vitro system as well as in an in vivo system. Having characterized the specific lysine residue that was involved in SUMOylation of RAN-GAP1, the researchers went on to investigate the cellular localization of both the wild-type protein as well as the mutated form of the protein, the one that was non-SUMOylatable. And it was rapidly established that the mutated form of the protein lacked the ability to become enriched at the nuclear periphery. This property suggested that SUMOylation was responsible for allowing RAN-GAP1 to associate to the periphery of the nucleus, potentially to the cytosolic side of the nuclear envelope. Subsequent studies allowed the researchers to establish that the SUMOylated form of RAN-GAP established close interactions with one of the components of the nuclear pore complex, specifically with one known as NUP358. NUP meaning nucleoporin 358, also known as the RAN binding protein 2. So for RAN-GAP1, SUMOylation is the type of post-translational modification that allows it to be localized in the cytosolic extensions of the nuclear pore complex, and that's the location where RAN-GAP stimulates the RAN GTPase activity of RAN, and therefore RAN-GAP plays a key role in regulating nucleocytoplasmic traffic within the cell, and that is all thanks to the fact that it is SUMOylated. The second example of a well-characterized SUMO target is IκBα alpha. IκBα alpha is the regulator of the transcription factor NF-κB. And NF-κB is the main regulator of innate and adaptive immune responses that occur within the cell. NF-κB is in fact activated by some of the signals that are transduced by the T cell receptor in T cells, but it also modulates the expression of pro-inflammatory cytokines in other immune cells. In the initial studies that led to the characterization of IκBα alpha as a SUMO target, it was established that the main form of IkBα alpha was very susceptible to be degraded by activation of the tumor necrosis factor receptor. In contrast, a higher molecular weight form of IkBα alpha, the one that was later characterized as being the SUMOylated form of IkBα alpha, was found to be very stable even upon activation by tumor necrosis factor. This led to the idea that SUMOylation of IkBα alpha stabilized IkBα alpha by preventing the polyubiquitination of IkBα alpha. This finding was further confirmed when the SUMOylation site in IkBα alpha was mapped, as it was found that the same lysine residue that mediated ubiquitination was the one that was being targeted by SUMOylation. So therefore, SUMOylation was competing with polyubiquitination, therefore affecting a protective effect on IκB alpha. However, the story got a little more complicated later on when it was established that besides being SUMOylated with SUMO1, IκB alpha could also be SUMOylated with some of the other SUMO paralogs, specifically with SUMO2 and SUMO3. And it was established that SUMOylation with SUMO2 or SUMO3 led to the formation of SUMO chains, in which a SUMO is then SUMOylated by another SUMO, producing chains that are similar in shape to those produced by polyubiquitination. Now, in this particular case, when IκB alpha was polysumoylated, those polysumo chains then led to the polyubiquitination of IkB alpha, therefore leading to the subsequent proteasomal degradation of IkB alpha. So, SUMOylation with SUMO1 was protective for IkB alpha, whereas SUMOylation with SUMO2 or SUMO3 was destabilizing for IkB alpha. And these findings led to the idea that SUMOylation can have multiple effects depending upon the paralog that is used for SUMOylation. And this is something that was not previously introduced, but it's important to keep in mind that in mammalian cells, specifically in human cells, there are 5 different SUMO paralogs: SUMO1, SUMO2, SUMO3, SUMO4, and SUMO5. And out of those, the ones that are expressed at very high levels, pretty much in all cells, are the first 3 paralogs: SUMO1, SUMO2, and SUMO3. And out of those, SUMO2 and SUMO3 are very similar to each other, whereas they only share about 50% sequence identity with SUMO1. The third example of protein SUMOylation deals with TDG, thymine DNA glycosylase, which is the first enzyme involved in base excision repair, a type of DNA repair mechanism that eliminates uracil residues produced by deamination events in DNA. The characterization of the effect of SUMOylation on TDG was started by identifying the specific lysine residue in TDG that was the target of SUMOylation. And that specific lysine residue that was identified as the main SUMOylation site in TDG was residue lysine 341. As substitution of that particular lysine residue for an arginine residue completely eliminated SUMOylation of TDG. The subsequent studies performed on SUMOylation of TDG were aimed at characterizing the shape of the SUMOylated form of TDG, and in fact this was one of the very first SUMOylated proteins whose structure became fully characterized. And upon completion of those crystal studies, it was determined that SUMOylation triggered a conformational change in the shape of TDG that destabilize its interaction with DNA. So therefore, SUMOylation of TDG triggered a conformational change that allowed for TDG to fall off from its target DNA. And from this perspective, it was obvious that in order to allow TDG to be involved in DNA repair once again, it was essential for TDG to become desUMOylated. And this is, in fact, achieved by the action of the SUMO isopeptidases. And the last example of the effects of SUMOylation on a protein target is one that is actually close to my heart, is on the effect of SUMOylation on the influenza A virus non-structural protein NS1. These were studies that were carried in my laboratory a few years ago. We were the very first group to identify NS1 as a target of the cellular SUMOylation system, and Upon the initial identification of NS1 as a SUMO target, our next goal was to identify the specific lysine residues that were targeted for SUMOylation in NS1. And those lysine residues were mapped to be residues lysine 70 and lysine 219 in NS1, with lysine 219 being the main target of SUMOylation in NS1 and lysine 70 being only a secondary SUMOylation site in NS1. Now, having characterized the main lysine residues that were targeted for SUMOylation in NS1, we also got to try to identify a mechanism to increase the SUMOylation of NS1. And for that, we developed something that we call an artificial SUMO ligase, or ASL, for NS1, and we were able to demonstrate that that artificial SUMO ligase increased in a very specific manner the SUMOylation of NS1 without affecting the SUMOylation of any other protein target within the cell. And with this tool at hand, this artificial SUMO ligase, we went on then to try to characterize the effects of SUMOylation in NS1 by studying the effects of doing both, decreasing the SUMOylation of NS1 by using the non-SUMOylatable mutant of NS1, as well as increasing the SUMOylation of NS1 by using the artificial SUMO ligase as a way to increase the percentage of SUMOylated NS1 within the cell. Quantitative analysis done by immunoblotting allow us to determine that the mutant form of NS1 was SUMOylated at less than 0.2%, whereas the normal form of NS1 was SUMOylated up to levels of about 1%, and in the presence of the artificial SUMO ligase, the total SUMOylation for NS1 reached levels of about 4%. And then we went on to assess the ability of NS1 to inhibit the production of interferon in the cell, because that's the main function of the NS1 protein of influenza A virus. And to do this, we use a transfection protocol that recapitulates the main events that happen during a normal influenza A virus infection, and by doing this, trigger interferon production by the same mechanisms that are normally involved in triggering interferon production in virus-infected cells. And in these studies, we were able to demonstrate that both increases and decreases in the total amount of SUMOylated NS1 are associated with decreases in the ability of NS1 to neutralize the production of interferon. This led us to postulate the conclusion that there is an ideal percentage of SUMOylated NS1 that is present in the cell during influenza A virus infection, and altering that ideal proportion of SUMOylated NS1 triggers negative effects on the ability of NS1 to modulate the production of interferon in the infected cell. One important conclusion associated to this idea is that SUMOylation is then the type of ideal cellular process that could be targeted for the development of innovative therapeutic treatments against influenza virus infection. While SUMOylation can be thought of as a post-translational modification affecting specific proteins within the cell, SUMOylation as a whole, as a cellular event, is known to modulate many processes that take place within the cell. Among others, SUMOylation is known to regulate the responses to cellular stress, as well as modulating nuclear integrity, modulating transcription, regulating chromatin structure and epigenetics, facilitating and regulating progression through cell division, and modulating cellular proliferation, acting as a main modulator of nucleocytoplasmic traffic, helping control cellular differentiation and stemness, and being a key factor in dictating progression into senescence and apoptosis, and also modulating innate immune responses and cell signaling. From that perspective, it is easy to see how simulation affects many outcomes in terms of the health of the overall individual. So simulation has actually been found to be key in processes such as neurodegeneration, carcinogenesis and metastasis, the regulation of autoimmune responses, responses to viral and bacterial infection, heart disease, and aging. And some of the most pressing questions in the SUMO field are related to the actual regulation, not only of the simulation of specific substrates, but also of SUMO substrates when they are present in large protein arrays, as it has been observed that there is mass SUMOylation of proteins within large protein complexes, and also the way in which the whole system can be modulated to trigger large increases or large decreases in cellular SUMOylation as a whole. And with this, we finish our introduction to cellular SUMOylation.